All right. We span more than 19 time zones. So let me start by saying good morning, good afternoon, and good evening, depending on where you are. My name is Adam Bogdanov, and as president of IS-MPMI, I want to welcome you to part two of the 2025 IS-MPMI Early Career Showcase. And for those of you who were able to join us last week, let me welcome you back. We have another exciting lineup of speakers this afternoon, or morning, or evening. And I don't want to stand between us and those very long, but I do want to introduce our moderators for today. First is Ved Prakash. Ved, can you wave? Ved is a postdoc at Kansas State University. And our second moderator is Ashley Nelson. Ashley, can you say hi? Ashley is doing her graduate work currently at North Dakota State University. And then I also want to introduce Katie Gruber. Katie is IS-MPMI staff. She's sort of running the technical side of the show, and she'll have some words for us at the break about the breakout rooms at the very end. So very briefly, we have 3 talks, then there'll be a short break, 3 more talks, and then we'll move into the breakout discussions, and we'll give you a little bit more information about those as we go along. That's all I have, unless I missed something essential, Katie.
You covered everything we need right now.
All right, then we'll hand it over to Ashley and Ved.
All right. Thank you. So without any further delay, we'll introduce the speaker. So we have Abigail Rogers with us, who is a graduate student in Purdue University, and he will be talking about the Ralstonia solanacearum effector protein U associates with and alters the organization of the plant cytoskeleton.
Okay. Everyone see my screen okay? All right. All right, good afternoon, everyone. Thank you so much for having me. I'm really excited to share with you all our findings from our recent publication titled "A Ralstonia solanacearum type 3 effector alters the plant cytoskeleton to promote bacterial virulence." As Ved mentioned, my name's Abigail Rogers. I'm a 4th-year PhD candidate in the Ira Pascuzzi lab here at Purdue University in Indiana, and I study effector biology and molecular plant pathology. My presentation today is going to follow this general outline. So first, we'll talk a little bit about the cytoskeleton and how it's a conserved target of multiple plant pathogens. We'll talk about the plant pathogen of interest in our lab, Ralstonia, and one of the type 3 effectors from Ralstonia that we identified, and how this effector RIPU targets the cytoskeleton to promote disease and alter cytoskeleton dynamics. Finally, I'll tell you what I told you in a summary of our findings, and we'll talk about some of the remaining questions and future directions this project will take. So without further ado, let's get into it. So as we know, the plant cytoskeleton is composed of both actin and microtubules. And when we think of the cytoskeleton, we really think about it in terms of promoting cellular structure, intracellular transport, including organellar movement. We know it plays a large role in secretory pathways, and we also know that it's responsive to both abiotic and biotic signals. So the biotic signal that we are primarily interested in is pathogen invasion. So upon pathogen perception and pathogen invasion, we see a restructuring of the cytoskeleton to form sort of physical barriers. As we can see here, at the site of a fungal aprosorium penetration, you see actin rearranging to sort of create a dense filamentous network. We also know that the cytoskeleton plays a role in the recycling and the positioning of pathogen recognition receptors, implying that the cytoskeleton has a huge supportive role in immune signaling. Additionally, we know that upon pathogen perception, the cytoskeleton is responsible for redistributing organelles intracellularly in order to sort of create a more site-directed increase in immune-related molecules. So given this role of the cytoskeleton in host immunity, it's no surprise that many plant pathogens target the cytoskeleton in order to induce disease, and they do so with secreted virulence proteins. Now, the pathogen that we're most interested in the IR-Pascusi lab, like I mentioned, is Ralstonia, which is the causal agent of bacterial wilt disease, the symptoms of which you can see here in this image. We have severe above-ground wilting symptoms. Now, Ralstonia is a broadly geographically distributed pathogen, and it also has a very broad host range. So given these two facts, Ralstonia is a really devastating pathogen, and in 2005, it was estimated that Ralstonia caused about $1 billion in yield losses worldwide. Now, Ralstonia infects upwards of 200 plant species, but some of the most nutritionally important include potato, tomato, and banana. And in the IR-Pascusi lab, we study Ralstonia solanacearum strain K60, which is endemic to the United States and therefore biologically relevant to us in Indiana. And this strain was originally isolated from tomato in North Carolina, so the system we like to work with in our lab is the Ralstonia tomato pathosystem. One of the major virulence factors of Ralstonia are these type 3 secreted effectors. These are virulence proteins, which from Ralstonia, these are known as Ralstonia injected proteins or RIPs, and they're secreted directly into the host cytosol through the type 3 secretion system, which is this molecular syringe. Now, in Ralstonia strain K60, there's a predicted 60 type 3 effectors. One of our previous graduate students in our lab, Rachel Hiles, was doing a screen of these effectors when she identified that one of them, RIP-U, seemed to co-localize with an actin-binding protein. So as we see here, in this channel, we've got RIP-U tagged with a green fluorescent protein, and here we have an actin-binding protein tagged with mCherry. And in our merged channels, everywhere that we see yellow is the overlap between the GFP and the mCherry signal. And this is really where our publication that I'll be talking about today kicks off. So at the time of this discovery, to the best of our knowledge, this was the first Ralstonia type 3 effector identified that appeared to co-localize with the cytoskeleton. So we really wanted to dig deeper into this effector. And to do so, we began by asking what the extent of this co-localization of RIP-U with actin was. So let's talk a little bit more about RIP-U and the cytoskeleton and both of their impacts on Ralstonia virulence and infection. So, like I said, our first question we were interested in was what the extent of this co-localization we saw between RIP-U and the cytoskeleton was. And to measure this, we used image analysis and we quantified the Pearson's correlation coefficient. And the Pearson's correlation coefficient essentially exists on a scale of +1 to -1. So you can imagine if you have two fluorescent channels, one where you've got a light purple fluorescence and another where you have a dark purple fluorescence. If you merge those channels and you see the two fluorescent signals perfectly overlay, this corresponds with a perfect positive correlation, and the inverse is true for a perfect negative correlation. Now, typically when we talk about Pearson's correlation coefficients, strong correlations are anything greater than plus or minus 0.5. And so this will be something that I'll bring up again and is something to keep in mind as we look at these images. So we moved from a laser scanning confocal microscope, which is where Rachel took her photos initially, to a spinning disk confocal microscope where we could get higher resolution to image individual actin filaments. And we see again, here's our RIP-U K60 tagged with GFP, and we have our actin binding protein tagged with our mCherry. And in our merged images, anywhere that you see white implies signal overlap. So that's what this yellow arrow here is pointing out. And as a control for these experiments, we used another type 3 effector from Ralstonia strain K60 called RIP-BD, which we knew localized to the plasma membrane. Now, when we go to quantify the extent of this co-localization, we see that our Pearson's correlation coefficient between RIP-U and RIP-BD is significantly different. However, our RIP-U is only weakly co-localizing with our actin marker, as its mean Pearson's correlation coefficient is still below this 0.5. When we went and looked at RIPU's co-localization with microtubules, however, we saw a much stronger association. So qualitatively, we can see a lot more regions of overlap in our merged channel compared to our actin. And when we look and quantify this and look at our Pearson's correlation coefficient, we see that this averages above a 0.5. So we could say that RIPU was strongly co-localizing with microtubules. However, microscopy data can only get you so far. Our next question was whether RIPU was actually physically associating with either aspect of the plant cytoskeleton. So although we saw that it was co-localizing, this does not tell us whether RIPU was directly interacting with tubulin or actin. In order to address this question, we first did a co-immunoprecipitation assay, and we saw that in plants expressing our RIPU tagged with GFP, we were able to pull down both actin and tubulin, which we were unable to do in plants expressing just our GFP tag. Additionally, in a yeast-2 hybrid assay with RIPU acting as both the bait and the prey, we saw that RIPU was able to interact with a tomato actin isoform relatively strongly. Additionally, when we followed up our co-IP with a mass spec, we saw that in RIPU expressing plants, we saw an increase or an upregulation of tubulin and actin in our samples. So this told us that RIPU was not only co-localizing with both actin and microtubules, but was also physically associating with actin and microtubules, which led us to our next question of whether RIPU is actually contributing to Ralstonia K60 virulence. Oftentimes, type 3 effectors will have functional redundancy, and so knocking out one of them might not actually influence the bacteria's ability to induce disease or colonize. So we wanted to know if RIPU was actually contributing to Ralstonia K60 disease-inducing ability. So what we did was we took our Ralstonia K60 wild type strain and we knocked out our RIPU effector protein, and we inoculated both resistant and susceptible tomato varieties and looked at the ability of Ralstonia to colonize over time. And what we saw was that our wild type Ralstonia, when RIPU was knocked out, we saw a significant decrease in the ability of Ralstonia to colonize. And notably, when we complemented RIPU back, we were able to restore that colonization ability. And this was something we saw consistently across resistant and susceptible tomato plants over time. But colonization is only one side of the coin, you could say. We also wanted to look at whether RIPU was actually helping to promote disease. So like I mentioned, the hallmark symptom of bacterial wilt disease is wilting. Now, I should note that although resistant plants are colonized by Ralstonia, they do not wilt. So here we basically have a control showing that, yes, our resistant varieties are resistant to wilting, induced by any of our Ralstonia strains over time. But notably, in our susceptible tomato lines, we see that in the RIPU knockout mutants, they're able to induce significantly less severe disease, so less severe wilting symptoms over time in our two susceptible tomato varieties. Sorry. So we knew that RIPU was physically associating with the cytoskeleton and playing a role in Ralstonia disease-inducing ability and colonization, but we wanted to know if cytoskeletal disruption generally was able to promote Ralstonia colonization. So we did a couple of assays. We pre-treated our tomato plants with latrunculin B, which is a chemical which binds globular actin to prevent actin polymerization. And we saw that by preventing actin polymerization, we could not only improve the ability of wild-type Ralstonia to colonize the plant, we could also restore the ability of our knockout mutant to colonize at the same level as our initial wild-type Ralstonia inoculated plants. And this was consistent over time for both resistant and susceptible tomato varieties tested. We also looked at microtubule disruption via pre-treating with the herbicide Arislin. And once again, we saw something very similar as we saw with actin. We saw that treatment of tomato plants with Arislin prior to Ralstonia inoculation improved the colonization of Ralstonia strain K60, where treatment also restored the colonization ability of our knocked out RIPU mutant as well. And again, this was consistent for both our resistant and susceptible tomato varieties. So we saw so far that RIP-U was needed for full Ralstonia virulence and pathogenicity. We also saw that chemical cytoskeletal disruptors could promote Ralstonia colonization. What we didn't know was whether RIP-U itself played a role in altering the cytoskeleton. So we went back to the confocal microscope and wanted to do image analysis on the organization of the cytoskeleton. And to do this, we quantified 3 different metrics. These include percent occupancy, which is a measurement of actin filament density. So here in the left-hand side, we see our fluorescent actin signals, and in our right-hand side, we see significantly more actin filaments upon treatment with Pseudomonas DC3000. Additionally, we wanted to measure the coefficient of variation, which is a measurement of actin bundling. So not only are we measuring the single actin filament density, we're also measuring the extent of these filaments coming together to create larger bundled structures. And finally, we wanted to measure microtubule number as well. And what we saw here was that at 48 hours, in plants expressing RIP-U, we were able to see a significant increase in actin filament density, so this percent occupancy, compared to our controls. And our controls here are plants expressing only our actin marker and our RIP-BD type 3 effector and our actin marker. Interestingly, however, we saw that there was no significant difference in our RIP-U treatments when we looked at the controls when considering the bundling of our actin filaments. Additionally, we saw that expression of RIP-U was able to significantly reduce microtubule number, as you can sort of qualitatively see in these images over on the left. When you compare the microtubule channel in RIP-U expressing cells versus in RIP-BD expressing cells, we do see visually less microtubules, and this corresponds with a significantly reduced microtubule number. So in summation, our findings from this paper include that RIP-U strongly colocalized with microtubules while physically associating with both actin and microtubules. We know that RIP-U is needed for the full Ralstonia K60 virulence and pathogenicity. And we also know that cytoskeleton disruption can promote Ralstonia colonization in tomato and also helps restore the loss of the type 3 effector RIP-U. Furthermore, we saw that RIP-U itself is having an influence over the arrangements of both the actin and microtubule cytoskeleton in planta. Now, some of the remaining questions we have include asking what is required for RIP-U virulence. Does RIP-U require an interaction with the cytoskeleton? Does it require disruption of the cytoskeleton in order to promote colonization and disease? What domains of RIP-U itself are actually responsible for both its interactions and its virulence function? And finally, we know that RIP-U is a core effector, which means that it's found in over 95% of all Ralstonia strains. So we're also curious as to whether RIP-U proteins from other Ralstonia strains function similarly to Ralstonia strain K60. And with that, I want to thank you all for your time and for having me, and I'm happy to answer any questions.
Great talk. So I see 2 questions in the chat. First is by Jingi. She's saying interesting work. Have you found any disruption of PRR or associated nanodomain by RIP-U?
That's not something we have looked at, but I think it's a really interesting question.
Okay. The other question is, great and engaging talk. Does RIP-U interact with actin and microtubules simultaneously, or does it interact with each one separately?
So we're also not sure about that. That's something that we're looking more into. So we're not really sure even if this interaction is direct or if it's mediated by some sort of middleman protein. So that is still something we're looking into.
The other question is from Adam. Your co-IP Western showed several bands of RIP-U GFP. Do you think that might be telling us something about how it works? That is processing of some short protein? Is it binding substrate and leading to substrate and self-degradation?
Yeah, so another really interesting question, and this is actually something we see pretty consistently in planta. We see that we get multiple bands whenever we do a protein pull down of RIP-U. So we don't know if it is interacting with something or if it's dimerizing or what's going on there. But again, something we're looking into for sure.
Okay, another one is from Jerry Lee. Great talk. Do you think if chemical disruption of cytoskeleton will also impact virulence of other pathogens?
If I had to, I mean, I would say probably yeah. Just the cytoskeleton has so many ways that it influences host immunity, that I imagine any disruption of the cytoskeleton would probably improve pathogens' ability to promote disease.
Okay, we have next question from Jean and Dennis. I would say let them ask.
Oh, hi. Very nice talk. I was just wondering if you could tell us with the RIP-U experiments, is this like chronically expressed RIP-U, or you have like how much does it mimic an infection the way you did the experiment? Like you pulsed it with RIP-U, or how long does it take to see the effects you're seeing?
Yeah,
How are you thinking about it?
okay. So I think if I'm understanding you correctly, kind of like what is our experimental design and how much is it biologically relevant to an actual infection? Is that like,
Yeah, just so like, do you think you're mimicking what happens during an infection, or, and also like the tissue type? Could you maybe talk about that? Like where
sure.
live you normally in the plant? Like,
Yeah, so Ralstonia is a root-borne pathogen, and all of our, I will say, sort of the caveat to these experiments is they are all done with transient expression. At least the confocal imaging is done transiently in Nicotiana epidermal cells, whereas Ralstonia is a root pathogen and invades the plant through the roots before working its way up through the vasculature to the stem. So I would say that our transient expression assays, like all transient expression assays, are not very they don't replicate an actual infection, right? But they are informative in telling us what RIP-U might be doing. When we look at that on sort of the timescale of an infection, that this is something we don't really know. We don't know when RIP-U is being expressed by the bacteria during infection, at what point, we don't know what cell type Ralstonia is in when it's expressing RIP-U. Those are just, we don't have that information yet. Does that answer your question? Okay.
Right. I'll try to be quick. So we published recently that infection with potato virus Y facilitates Ralstonia infection in potato, and that's presumably due to a reduction in callus production from HC Pro of potato virus Y. Have you looked at any responses that might be facilitated by actin filaments or the cytoskeleton, like callus deposition? Does RIP-U deletion increase callus that could reduce its virulence?
Yeah, so something we're working on, and we had preliminary data for, is we're working on generating stable transgenics that can inducibly express RIP-U. So then we can challenge those with PAMP in order to look at things like callus deposition. So our qualitative data, which is very preliminary, I should say, suggests that when RIP-U is being expressed and then you treat with a PAMP on top of it, you do see, again, qualitatively, less callus. This is something we want to continue to look at a little bit more. So short answer, yes, that is something we're interested in.
Very cool. I should have led off, but great talk. Yeah.
thank you.
All right. So our next speaker is Elaine Rim, a postdoctoral scholar from University of California, Davis. And her talk is Directed Evolution to Engineer Immune Receptors that Recognize Novel Pathogen-Derived Effectors. Back to you, Ellen.
Thank you for the introduction. My name is Ellen. I'm a postdoc in Pam Ronald's lab at UC Davis in the Department of Plant Pathology, and I'm a plant synthetic biologist. engineering proteins to enhance pathogen resistance and climate stress resilience. So today I will be telling you about directed evolution and the disease that we're targeting there, and tell you about engineering an immune receptor through that process, and then testing this engineered immune receptor in planta. And briefly, I'll tell you about another way to leverage directed evolution data. Then I'll end with a conclusion. So before we get into the directed evolution part of this, I wanted to talk about why we want to engineer immune receptors in the first place. So we know that plants and pathogens are locked in this competition where pathogens secrete these molecules called effectors, and that they help pathogen infection, whereas plants have evolved these immune receptors that can recognize specific effectors, and when there's that binding interaction, you get immune response that protects the host plant. But we also know that pathogens are very good at evolving such that their effectors escape detection by these immune receptors, and then there's no binding, then no immune response gets activated, and the plant host is now susceptible to the pathogen. So knowing how this competition works, the overarching goal is, can we speed up evolution of immune receptors to help plant hosts? And here I'm showing you the seal for Operation Warp Speed, which led to great mRNA vaccines that helped us throughout the pandemic, which we're not making vaccines here, but the principle of tipping the scale in favor of a host against a fast evolving pathogen by taking unusual measures is something that I think we can compare to. And that unusual measure in our case is directed evolution, which in a nutshell is a way to evolve useful immune receptor variants in a lab setting. And this involves screening many variants of an immune protein to find a desired new function. I'll go into more detail about this process later, but I wanted to tell you about the disease that we targeted. Rice blast is a disease that's caused by the fungal pathogen Magnaportha oryzae. This is a devastating disease, especially in humid climates around the world. And I'm showing you here pictures of a picture of diseased leaves in rice. And this disease can lead up to annual loss of rice enough to feed 60 million people. So the engineering target for me was an immune receptor in rice called PIC1. This receptor binds the effector AVR-PIC secreted from Magnaportha oryzae. The specific allele that I targeted is the PIC-H allele. And this is one of the best functional alleles in rice, and it recognizes the AVR-PIC-A variant of the effector, but it still cannot recognize AVR-PIC-C or AVR-PIC-F variants, which are only different by 1 to 3 amino acids. So the fungal isolates that only express AVR-PIC-C or AVR-PIC-F would escape detection by this best version of the natural receptor. So knowing this interaction, the goal for me was to engineer PIC-H1 variants that bind both AVR-PIC-C and AVR-PIC-F, and down the road be able to provide the broad spectrum resistance against Magnaportha oryzae isolates. So that was the goal I started with. In terms of how to get there, I wanted to engineer PIC-H1 using a high throughput screen of PIC-H1 variants for this new to nature binding ability. And so what I mean by that is, if I test these genetic variants in individual plants, even if I spend a lot of time in the greenhouse, that would only mean hundreds of genetic variants in a single experiment. So I wanted to increase the throughput and speed up this process by testing the receptor variants in individual cells. And this would lead to tens to hundreds of millions of genetic variants screened in one experiment. And to do this, I used single yeast cells, which you see in the picture on the right here. So the question then becomes, how can we recapitulate plant immune receptor function in these single cells? And I've used yeast surface display as a way to do this. And in this technique, you can express the effector binding domain of your receptor on the surface of single yeast cells. And that receptor expression can be detected through a fluorescent signal picking up an epitope tag. And that is detected in magenta in my images, as you see here with the PIC-H1 expression. So these circles around the yeast cells indicate that the receptor is expressing properly on the cell surface. At the same time, you can fluorescently label your effector, so AVR PIC, with another fluorescent signal and detect binding or not binding. So here I'm showing AVR PIC A in cyan going and binding the PIC-H1 effector binding domain at the cell periphery. So using this two-color imaging, you can look at both your effector expression and effector binding. So that's AVR PIC A binding, but as we expect, AVR PIC C and AVR PIC F, which are known to not interact with PIC-H1 receptor, does not show cyan signal where the receptor is, indicating that this technique recapitulates what happens in rice. So that was good to see, and I could move on to perform the directed evolution platform. So this starts with generating a large collection of variants of the receptor that you're trying to engineer, and that's done through random mutagenesis normally. And these variants are introduced into single yeast cells for expressing on the cell surface. And these cells are then, they undergo selection based on fluorescence because fluorescence correlates with effector binding. And this selection process happens through FACS or fluorescence activated cell sorting, where you can quantify the fluorescence of single cells. So after you've selected the variants that have gained binding to your target effector, you sequence those variants through high throughput sequencing, and that tells us which variants have gained binding. And it's important to take these variants back into plants to test their function in model and crop plants. So that is the directed evolution cycle that I applied to the rice PIC-H1 receptor. So I started with random mutagenesis through error-prone PCR to generate 20 million receptor variants that were expressed on the yeast cell surface. And then I subjected this population of variants to a series of selection events or sorts, and the details are not as important, but I alternated between AVR PIC C and AVR PIC F with the goal of finding receptor variants that have binding for both effectors. And what I mean by selection round or sort is something like this. This is an exemplary FACS plot where on the x-axis is receptor expression that is quantified through one fluorescent signal, and the effector binding is quantified on the y-axis through another fluorescent signal. And each dot here indicates a cell expressing a receptor variant. So what I selected were variants in a window like this, where these red dots indicate cells that have good receptor expression and higher effector binding as they are shifted up on the y-axis. So I ended up selecting top 0.5 to 1% of the variants in each selection round. So after this series of selections, I was able to recover some variants that had improved function. And I'm showing you one such variant that I called A4.1. So in magenta, we see again the receptor expression on the cell surface. And when it comes to effector binding, it retains AVR PIC A binding, as you see in the top image, and it has now gained binding to AVR PIC C and AVR PIC F that you see in cyan. And to go back to the PIC-H1 receptor for comparison, it does not show binding for AVR PIC C and AVR PIC F. So we went from being able to recognize AVR PIC A, but not these other effectors, to being able to pick up all these variants. And then I wanted to test the function of this engineered immune receptor in planta. And to do that, I used Nikushana bentamiana cell death assay, which a lot of you are familiar with. In this assay, you introduce the immune receptor and effector into Nikushana leaf tissue using agrobacterium infiltration. And when there is binding interaction between the two components, you see cell death that is marked by yellowish color in our images under UV light. When there's no interaction, then there's no immune response, and that tissue remains intact. And this was done in collaboration with my lab mates Oscar Garrett, Yejin Shim, Jack Carter, as well as our collaborator in the Dinesh Kumar lab at UC Davis, Yeon Lee. So if you look at this leaf, on the left side, we have our negative control YFP, which should not induce cell death, and that's what we see. And the wild-type PIK-H1 receptor in response to AVR-PIK-C also does not induce cell death, whereas our engineered receptors A4.1 and B4.1 does have strong cell death. Similarly, with AVR-PIK-F, we do not see cell death response with the wild-type receptor, whereas A4.1 and B4.1 is able to induce cell death. So that's quantified across 3 experiments here. And I'll note that this assay has a lot of plant-to-plant variability, but across these experiments, we had the engineered receptors showing AVR-PIK-C and AVR-PIK-F recognition over the wild-type receptor. So that was a new-to-nature function that we engineered through directed evolution. And I actually wanted to push this process a little further and engineer the PIK-H1 receptor to bind another effector called AVR-PIZT. And this is another Magnaportha-Oryzae effector that is sequence and evolutionarily divergent from the AVR-PIKs. And through additional selection rounds, we were able to gain binding to this other effector. And in the interest of time, I won't show that part of the story, but the what I've shown you so far in the AVR-PIZT story is now on bioRxiv if you'd like to take a look. So we have these engineered receptors that were tested in tobacco plants, but we are now testing the function of these receptors in rice. So we've introduced our engineered receptors into rice through agrobacterium transformation as transgenics, and those plants are growing now to collect seeds and inoculate the seedlings with different Magnaportha-Oryzae isolates with the effectors that we targeted. So that's to test the function of our receptors initially, but the goal down the road is to be able to introduce these mutations that led to enhanced function using CRISPR editing. So I'm showing you here the sequences of the effector binding domain of the variants that I've told you about, and they have 5 amino acid substitutions along the stretch. So in the future, it would be possible to introduce these substitutions via prime editing or base editing into the endogenous immune receptor gene. And that would be the goal moving forward with our receptor engineering approach. But that still requires engineering. What if we can identify natural variants of immune receptors that are functional and introduce those into our cultivars through breeding right away? And data from directed evolution can actually inform such efforts. So when I sequence these variants that I select for after binding interaction assay, I go through high throughput sequencing, and that gives us data like this, where you have the variant sequence of the effector binding domain on the left, and the read count is shown on the right. The reads that's mapped to this particular sequence. And the read count roughly reflects the binding function. So the sequences that are more highly represented indicate greater binding to the target effector in general. So you have this data very rich in sequence function information, and that can actually be used to predict the function of naturally evolved sequences through machine learning approaches. So that is another effort in the lab in collaboration with computational biologists Alex Howard and Jim Notwell. And that story is also on bioRxiv if you'd like to take a look. So to summarize what I've told you today, the rice immune receptor PIK-H1 was engineered through directed evolution to bind previously unrecognized effectors to gain this new-to-nature function. And this function is largely recapitulated in our Nicotiana bentamiana assays. We're testing the engineered receptors in rice to see whether they can actually protect from previously unrecognized isolates of Magnaportha-Oryzae. And lastly, I've told you that data from directed evolution can train machine learning models to predict the function of natural variants, which can then be used for breeding efforts right away. So with that, I'd like to thank Pam, my advisor, as well as my lab mates highlighted here who have been part of the project, and our collaborator Yuan Yuan, and my funding sources. I'd be happy to take any questions.
All right, thank you, Ellen. Great talk. Let's see. I don't see any hands up, but I know I have a couple of questions. So, let's see. We might have one from the chat first. Let's see. Okay. So, one of my questions is, for the system, do you think it would ever be adaptable to effectors that aren't directly interacting with the resistance gene, or is it based on only direct interactions?
Yeah, that's an interesting question. We'd have to find a different readout for that. You know, if there's a plant component that gets modified, is there a fluorescence readout for that that we can use in single cell systems? I feel like without direct interaction, it would be harder to apply to single cell systems because you might need a lot of other plant components to recapitulate that readout. But yeah, so far we are only working with direct interactors.
Okay, thank you.
I could go ahead. Yeah, so great talk, Ellen.
Yeah, that'd be great.
And just the new approach to bind a more distantly related effector, do you change your strategy in any way in terms of like the kind of intensity of the mutagenesis or the thresholding? I'm just thinking, how far can you get with like a single round? Yeah, versus these more closely related, basically alleles.
Yeah, thanks, Adam. That's a really good question, and that's really so it is harder to push them to bind more distant effectors. So my strategy was subject the outcome of the first couple rounds of selection against AVR-PIC-C and AVR-PIC-F through additional rounds of selection. But at that point, the diversity of the library was so depleted that we actually had to introduce more diversity through another round of mutagenesis. So we were able to find the variants at the end of 7 rounds of selection total and 2 rounds of mutagenesis, a couple variants that had binding to AVR-PIC-C, AVR-PIC-F, and AVR-PIZT in the yeast assay, but translating that to plants, to all of the effector binding with the strength that we see in yeast to plants has been difficult. So I think there's something about the effector binding orientation maybe that's different between these classes of effectors. We also have to take that into consideration. So I think to target something more distant, we do need a lot of diversity, and it's an interesting question whether serial selection or parallel selection might be better. But yeah, I think it's potentially applicable to other divergent classes. Thanks.
All right, so we have a few questions from the chat. So Roger says, really nice work, Ellen. Does AlphaFold make any clear predictions on how these substitutions affect the binding to the variant effectors?
Yeah, that's a good question. I haven't shown you what the mutations are. It's with all 3 of them, but the AlphaFold does okay. I don't think it's possible to predict exactly which residue is responsible. We did do some assays where I went in and mutated 1 amino acid at a time, and it seemed to be a combinatorial effect. And even the residues that are outside the interaction, the domains of this receptor, which is known. So I think the AVR-PIC pair is kind of unusual in that we already have a crystal structure, and we can probably learn more from the crystal structure and going in and mutating those residues and see what happens. There's actually one mutation that I think a lysine to isoleucine that I think makes it possible for our variants to bind more broadly without that dangling lysine residue. So we are doing some biochemistry. We haven't seen anything obvious from predictions.
All right, so we do have a few more questions in the chat, but we're running out of time. So I'm just going to ask Ellen to respond to those in the chat box. I'm going to hand it over to Katie.
Wonderful. Thank you, everybody, so far. Yes, it's been a wonderful first half of our session here. So we are going to move into our break. We have 10 minutes for you to stretch your legs or grab something to drink. I do want to mention that later, after we come back from our short break here, we will be moving into breakout rooms. So these are some of the topics. I will address these later. Again, you do not have to memorize them right now. But these are the breakout rooms that we will be able to choose from to attend and have some great discussion. We also want to remind you that the IS-MPMI Congress is happening this summer in Cologne, Germany. And an abstract submission is open until March 11th, and registration opened this past week. If you have any additional questions, feel free to scan the QR code and you can review the program or any hotel information that has been posted. And then finally, as we are halfway through our session here, we just want to say thank you again for your participation. And with that, we will start our 10-minute break. So we will come back at the 18 mark with our second half of our presenters. Thank you very much. All right. Hopefully everybody enjoyed the short break here. And I am going to turn it back to our moderators to kick off the second half of our second session. Thank you.
All right, thank you, Katie. So I'm going to introduce our first speaker, Jian Chen, who's a postdoc at Cicero in Canberra, Australia. And today he's going to be talking about effector recognition and activation mechanisms of NLR kinase fusion proteins. Jian, are you able to share your screen? Sorry, is everyone able to hear me okay?
Yes.
We
Okay, so I
can.
think
Yeah.
okay, I think with that, we're just going to pivot to Neftali, if you're okay presenting first today, or presenting first in our second half. So Neftali is a postdoc at the Sainsbury Lab in Norwich, and today he's going to be talking about the phosphorylation landscape of rice blast disease.
Hello, everyone. So thank you very much for the opportunity to present my work today. So I am Neftali Cruz Mireles. I'm originally from Mexico. And until a few weeks ago, I was at the Sainsbury Lab in Norwich. But now I'm at the University of Zurich, where I'm going to briefly mention what I'm doing here at the end. But I'm a postdoctoral researcher, and mainly my main interest has been related to phosphoproteomics and how phosphoproteomes change during plant microbe interactions. So this talk is going to be, again, talking about rice blast disease, in particular about the pathogenicity map kinase PMK1. And also, I'm going to show how we have used phosphoproteomics to identify novel virulence determinants. And then I'm going to show you an example of this data set that we have been investigating a novel molecular mechanism of rice blast to cause disease. And at the end, I'm going to wrap up with some key take-home messages. So as I mentioned, I have been particularly interested in protein phosphorylation in signaling, and this is because this is an important post-translational modification. So with phosphorylation, we can activate or deactivate proteins, but also we can modify interactions between two proteins as well. At the same time, this post-translational modification can change the localization of some proteins, or it can also mediate or regulate the stability of some other proteins. So I have been investigating this during rice blast disease. Already Helen did a very nice introduction of this disease, but I just want to remind you that rice blast is an important disease because it infects more than 50 different grass species. And basically, this disease causes this type of lesion in grasses that once the disease started, so the crop or the cultivar is gone. So this disease starts with a conidia cell that lands on the hydrophobic surface of the leaf. That then, after a few hours, this spore germinates to generate this germ tube that attaches to the leaf surface. And then later on, after 24 hours, this spore collapses by a process called autophagy, and then leads to the generation of this novel structure called the apresorium that will allow the pathogen to penetrate the leaf and cause the infection. So basically, in our lab, so we were very interested in the pathogenicity map kinase PMK1 because we know it's an important pathway from the fungus, from the blast fungus, to cause infection. So we know that this map kinase is, as other map kinases, is activated by an external stimuli that as soon as this signaling is activated, it will then phosphorylate the map kinase PMK1. And this phosphorylation will activate or will mediate processes such as conidiation, appressor information on penetration in the blast fungus. So we know this from 20 years already of research in this MAP kinase signaling pathway. But there are still like many open questions. So although we know that this signal is important for different developmental stages in the blast fungus, but still we don't know what are the downstream targets or the full components of this MAP kinase signaling. And with this, I started my PhD project. So basically, I decided to investigate the phosphoproteome of the rice blast disease during appressorium formation. And basically, I did this by comparing the phosphoproteomes of the wild-type strain G11 and the null mutant of the MAP kinase PMK1 that is unable to form an appressorium. So just to show you this, basically, when we infect rice blast wild type, we can see this type of lesions that I showed you earlier. But in the case of the null mutant of the MAP kinase, we don't see, it's non-pathogenic. We don't see any lesion. So then I decided to investigate the phosphoproteomes of these two strains during infection-related development, and then with the idea of identifying some important phosphorylation sites for in different proteins for important for infection. So with this, we generated what we call the global phosphorylation landscape of the appressorium in Magna Portorosa. So we did this in a time course dependent manner. So basically, we germinated spores of the wild type strain or the null mutant of PMK1 in hydrophobic cover slips. And we did this from 0 to 6 hours. And then, well, this was a lot of effort just to generate this material that then later we used for phosphopeptide enrichment. And with this analysis, we were able to identify around 8,000 phosphopeptides important for or participating in this early infection process. These phosphopeptides were corresponding to around 2,000 proteins or phosphoproteins. And then when we cluster this information, so we realized that there is a group of highly phosphorylated proteins only during the spore stage. So then we also identify some cluster of proteins that are highly phosphorylated during germ tube formation or during early germination. And later on, once the appressorium of the Magna Portorosa fungus is formed, so we can see that there is a high number of phosphorylated proteins at 4 and 6 hours of germination. So with this information, so then we generated this resource that we wanted to use to investigate or to identify novel virulence determinants in the rice blast fungus. So I'm just going to show you here some or mention some examples that we are now investigating in the lab. And basically, we know that this phosphorylation data that we have, we can identify different phosphoproteins that we know are important or might be important for pathogenicity. And for instance, we identify some phosphosites of proteins involved in the MAP kinase signaling pathway, including some transcription factors and some other kinases. But also in other processes such as autophagy, a process that I mentioned earlier is important for the spore to collapse. So we know that also there are some phosphorylation sites that are important in some processes, especially at the initiation of this autophagy pathway. So we are currently investigating some of these phosphosites and the relevance of these phosphosites in Magna Portorosa. But the example that I'm going to show you today is this protein called BTS1. So this BTS1 is a phosphoprotein that we identify, like it has two different phosphorylation sites, one in the serine 175 and one in the serine 420. And basically, when we knock out this gene in Magna Portorosa, we can see that the knockout has this phenotype in germination. So we have some aberrant germination, forming this kind of secondary structure here in the germ tube. But also the null mutants of BTS1 have a reduced infection compared to the wild type strains. So these two phenotypes can be complemented when we introduce again the BTS1 allele in the null mutant of BTS1. So then we also wanted to see whether this protein BTS1 interacts with the MAP kinase PMK1. And when we pull down BTS1, we can observe that in these pull downs, we can see PMK1 or the active version of PMK1 interacting or associating to BTS1. Also, we produce recombinant proteins of BTS1 and PMK1 to do an in vitro phosphorylation assay, and then we observe that only when we activate PMK1, we can see phosphorylation of BTS1 in serine and threonine residues. So with this information, we found that indeed PMK1 associates with BTS1 and can also phosphorylate this protein. So to understand the relevance of these phosphosites in the context of virulence, we then did phosphomimic and phosphodead mutations in these phosphorylated residues. And basically, we just did alanine substitutions to generate a phosphodead version of the allele. And also we did a phosphomimetic substitution with aspartic acid to mimic the phosphorylation in these residues. So what we observed is that out of the two phosphosites that we identified, the serine 175 had a kind of similar phenotype in the phosphodead comparing to the wild type. But then in the case of the phosphomimetic version was more similar to what we see in the G11 wild type version. So suggesting that this phosphosites is important for virulence. So in the case of Presorium formation, again, we assess these phosphomutants and then observe the phenotype. And what we observe again is that in the case of the position 175 mutations, again, in the phosphomimetic allele is more similar to what we observe in the wild type, and then the phosphodead is more similar to what we see in the null mutant of BTS1. Then suggesting that this phosphosite is required for Presorium formation, but also for virulence of the blast fungus Magna portoroica. So then we wanted to understand the function of these proteins in the context of rice blast disease. And for this, we did a localization study fusing BTS1 to GFP, and we observed that this protein localizes at the base of the Presorium Magna porte. And actually, when we use cytoskeleton markers such as septins, we can see that this localization of BTS1 is very close to the Presorium pore that I'm showing you here. And the Presorium pore we know is important for the penetration of the fungus in the host. So then we basically think that these two, or BTS1 localizes close to the Presorium pore to have a function in this particular site. And later, we also found that this phosphoprotein BTS1 has a sterile alpha motif, or called SAM domain, that we know is important in other signaling pathways to modulate protein-protein interactions. So we investigated this in more detail, and then for this, we used some orthologs of BTS1 in Saccharomyces cerevisiae, where we know this SAM domain is important for to mediate RNA protein RNA binding interactions. And also, this protein is ortholog of the SMOG protein in Drosophila melanogaster that also has a SAM domain. So when we overlap the SAM domain of these two proteins associated to, or that have been related to RNA binding with our protein from Magna portoroica, we can see that these SAM domains are pretty similar. And actually, when we align these sequences, we can see that the residues that have been reported to have a role in RNA binding are highly conserved in between these three SAM domains. So then we wanted to know whether the function of the BTS1 in GIST can complement the function in Magna portoroica. And then for this, we complemented this GIST allele in Magna porte, and we observed that the localization is, again, very similar to what we saw before at the base of the Presorium. And then when we complement this allele in the null mutant of BTS1 in Magna porte, we can see that it can also restore pathogenicity. Something that we also did to investigate the function of the SAM domain of this protein in Magna portoroica is that we did a truncation of the SAM domain. And with this mutant, we observed that also an aberrant germination of the germ tube, forming these very, very long structures that have again a similar localization to the wild type in the upper sodium. So, however, this truncated version of the SAM domain is not able to complement the pathogenicity function of this protein in rice blast disease. So finally, to investigate more of the function of this protein during infection, we did an interactome of BTS1 during 0, 4, and 24 hours of infection. And what we observed is that mainly the interactors that we found in these 3 time points are different, but also some of the top interactors of the top candidates from this assay are proteins related to RNA degradation or cytoskeleton components. So then we are at the moment investigating the association of BTS1 with these different proteins, but it's exciting to see these RNA degradation proteins. So just to summarize, basically we use phosphoproteomics to investigate early upper sodium formation in the blast fungus. So we use a comparative approach to define the phosphoproteome of this infection process. And then we investigated one of the proteins called BTS1 that we know interacts and is phosphorylated by the MAP kinase PMK1. So when we wanted to know a bit more about the function of BTS1, we know that this is a cytoplasmic protein that localizes at the upper sodium. So it's a protein that is predicted to bind RNA. And this protein also associates to RNA degradation proteins with the IPMS results. So with this, what we think is that the function of this SAM domain containing protein BTS1 associates with PMK1 during upper sodium formation and penetration, and potentially is bringing together some RNA that are important to localize at the upper sodium pore for the function of this mRNA during infection. So with this, I'd like to thank everyone in the Talbot lab and also in the proteomics team at the Sainsbury lab, which helped me a lot with this project. And as I said, so I just recently started now a new postdoc position. So now with an EMBO fellowship in Zurich at the University of Zurich with Professor Cyril Zifril and Professor Pedro Beltrao, where I'm going to be investigating now phosphoproteomes in Arabidopsis or in plants, also in response to different biotic stresses. So with this, I'm happy to take any question.
All right, thank you, Neftali. Let me just see if anyone's raising their hand. I can start us off with a question. So knowing now this VTS1 that you got through your phosphoproteomics, do you think some of the data you're getting from that you could learn, you could almost predict maybe some important phosphorylation proteins?
Yeah. So, well, we are, as I said, still exploring the data set. So BTS1 was one of the first proteins that we are kind of trying to investigate the molecular mechanism and how phosphorylation is important. But yeah, there are other proteins that potentially are associated to this MAP kinase signaling pathway. And we think that they might be also forming complexes with BTS1. And perhaps this protein might be regulating some of the signaling that are being transduced through this signaling cascade PMK1. So, but yeah, there might be, or we think that there are also other phosphoproteins that are related to these proteins during rice blast infection.
Great, thank you. Okay, so Roger is saying, nice talk, Neftali. Is VTS1 secreted from the fungus? Related to that, were those interacting proteins fungal or plant proteins?
Yeah, so all of the proteins that we identified in our phosphoproteomic study are fungal proteins. So we did this was an in vitro infection, so using hydrophobic cover slips. So then all this, we also tried to do the same assay in planta, but obviously the phosphoprotein from the plant was a lot. So we were more interested in what was happening in Magna porteriaceae. So then that's why we decided to do it like in vitro. And we don't think BTS1 is secreted. So we don't have any, there is no any signal or any peptide that might relate it to secretion. Or also, we know that the localization of the protein is just mainly in this upper base of the upper sodium. So we think that, this protein might not be secreted. But so then the proteins that we think might be interacting with BTS1 will be all fungal proteins.
Great, thank you. And then Jean has her hand raised.
Very nice talk. Because I like to complicate things, I want to know if you also search for other PTMs, and how, if they were changing or interfering or doing anything interesting.
Yeah, so something that I didn't mention is that the protein that we investigated, so we enriched for phosphoproteins only. So then that's why we only investigated this modification. And also something that we know at the moment, we can only identify serine and threonine and tyrosine modifications. So we know that there might be other phosphorylation in other residues that we haven't detected. But yeah, so it will be nice to see, I don't know, other type of modifications. I know that there are some groups studying summoylation, but I think at the moment there is not much information about other modifications, at least during rice blast infection.
But you didn't see anything like, were you enriching peptides or whole proteins? How did you?
Yes. So basically, we enrich for phosphopeptides only to do this analysis. So then we didn't we were not interested in checking other modifications. So the peptides that we got were only enriched on phosphorylation for phosphorylation. Yeah.
All right, thank you. If you guys have any follow-up questions, just ask them in the chat and Nestle will get back to you. And then I'm going to introduce our next speaker, which is Jian Chen, and he's joining us as a postdoc from Cicero in Canberra, Australia.
Thank you. And sorry, I'm late to come back in the Zoom. Thanks you very much for having me here to present our work on SR62, which is a weight tandem kinase immune receptor, and it can activate an NRR to trigger immunity. I'm a postdoc from Australia Commonwealth Scientific and Industrial Research Organization. And today I'm going to talk about the identification of the AVR SR62 effectors, and then how does the SR62 tandem kinase protein and the NR protein function together, and then show some beautiful predictive structures. Okay, first is the introduction. Rust fungi are very harmful disease for plants, and our group called the Pests and Disease Group. We are working on many weight rust diseases like strip rust, leaf rust, and stem rust. We have been identifying weight resistance and understanding rust dynamics and identifying evidence genes for many years. So many weight resistant genes and rust effector genes have been identified by our group. And another very important work that is by us is developing durable resistance. So for this work, apart from the markers for breeding, we are also making cisgenic and transgenic gene cassettes that will provide durable resistance. So understanding how immune receptor or R gene function is very important to guide the deployment of resistance. Most of the cloned R genes in wheat are NRR immune receptors. Generally, CCNB proteins, and NR proteins have been well studied especially in recent several years. They can recognize the effectors that are secreted into plants by pathogens and form resistance to execute immune response, and normally with cell death at the infection site. Many recent cloned R genes from wheat are encode kinase fusion proteins. They are not rare because nearly 20% of cloned R genes belong to this new class of immune receptors. For example, SR62, which is a stem rust resistant gene, it has two kinase domains fused together. So as this class of immune receptor are not rare, they are pretty common, 20%. So understanding how this kind of immune receptors function is very important for the utilization of them. So for us, our purpose is to understand how does the SR62 tandem kinase protein function to confer resistance. The first step to start the story is identification of the cognitive effector of SR62. We use the purposed screen method to identify effectors. This is a very good method developed by our group. So using this method, we screened the stem rust effect library and get 4 effector candidates. And these 4 effector genes located in complex locus in chromosome 5A and 5B. And apart from these 4 variants, there are another 3 variants in this locus. So 7 in total. When we co-express these 7 effector genes with SR62 tandem kinase protein into weight purposed, and 4 of them showed strong cell death. This means these 4 are the recognized evidence effectors, and another 3 not. So this means the other 3 are the non-recognized valence effectors. But when we co-infiltrate these effectors with the kinase protein in nicotinamide benzoyl, we cannot see any cell death. So this means other proteins are required for their function in the system. So this conclusion lead us to the work from our collaborator, Brandon Wolf team from COST. From their work through mutagenesis screen, they find that there is a linked NRR gene in the SR62 tandem kinase gene locus. And this NRR encodes a CCNB protein, but with 2 NB domain. This is very rare. And when we get this NLR, we can infiltrate it with the SR62 effectors and the tandem kinase protein. And you can see that 4 of them induced very obvious cell death. And this cell death pattern is consistent with the previous purpose data. So this means that the effector and plus the kinase protein and the NR, these 3 components are enough to activate immune response. So the question is how do these 3 components function together to confer immune response? To answer this question, we start from detecting the relationships, the interactions between these 3 proteins. For example, we tested the interaction between the effector and the kinase protein and the effector with the NR protein. We used to hybrid co-IP split and planned to hybrid a series of methods to detect the protein-protein interactions. Here, I only show the result from planned to hybrid, which is the split Gopher Ruby assay. This is also a very good method developed from our group. And generally, we move the yeast to hybrid from yeast to plant, but use the Ruby as the report gene. So you can directly see the protein-protein interaction from the infiltrated leaf. From this leaf, you can see that 4 of the effectors show interaction with SR62 tandem kinase protein, and the other 3 effectors not. And this interaction pattern is consistent with cell death. And we observed a similar result from the other method. So this means SR62, this tandem kinase protein, takes the role to recognize the effectors. As SR62 tandem kinase protein have 2 domains, K1 domain and K2 domain. So the next step, we divide the domain into 2 separate domains, the protein into 2 separate domains, and tested its interaction with the effectors. And only kinase 1 domain showed interaction with the effectors. So it is actually the kinase 1 that is responsible for recognition of effectors. So what is the function of kinase 2 domain? When we co-infiltrate the NLR with the kinase valence protein, kinase 1 domain, and kinase 2 domain, we find that kinase 2 domain itself is enough to activate the NLR without effector, which means it is autoactive. But the SR62 tandem kinase protein, valence protein, must need to recognize the effector first and then can activate the NLR. So why cannot this valence kinase protein directly activate the NLR? This question can be answered by the following plant 2 hybrid assay. You can see that from this plant 2 hybrid assay, kinase 1 domain interact with kinase 2 domain. But when you co-infiltrate kinase 1, kinase 2 with evidence recognize the effectors, you cannot see the interaction between kinase 1 and kinase 2 anymore. And as control, if the co-infiltrated effector is non-recognized variance effector or just a YP, K1 still interact with K2. So this means recognition of the evidence AVR SR62 effectors will suppress the interaction between K1 and K2. And this interaction and suppression relationship can also be reflected from the cell death assay. From this leaf, you can see that K2 can activate the NLR and cause strong cell death. But when you co-infiltrate K2 and K1 together, they cannot activate the NLR. And if you co-infiltrate K2, K1, but plus a valence effector, then they can activate the NLR and cause strong cell death again. And if the co-infiltrated effector is the non-recognized effector, they cannot activate the NLR. So this means the recognition of the valence effector can release kinase 2 from the inhibition and then activate the NLR. This conclusion can be further demonstrated by the following co-IP. You can see in this co-IP, the kinase 2 domain interact with the NLR, but the kinase 1 domain not. And the valence protein also have weaker interaction with the NLR protein. And this weak interaction between the kinase valence protein and the NLR can be further increased if we put a valence effector in the system. And as control, if we put the non-recognized valence effector or other effectors, they cannot increase the interaction between the kinase protein and the NLR. So it means the recognition of the valence AVR SR62 effector will promote the interaction between the kinase protein and the NLR. So based on the previous results, the above results, the model for this work it will be that SR62 tandem kinase protein stay as self-inhibited state by interaction between K1 and K2, and the AVR SR62 effector will be recognized by binding to the kinase 1 domain, and the binding of effector will push K2 away from inhibition, and then it can interact with NLR and activate it. So how exactly this process happens, how these proteins interact and function? And this question will lead us to the work structure prediction section, which was performed by Stephen team from COST. You can see that from the predicted structure, SR62 tandem kinase protein stay as a dimer by self-association, while the kinase 1 domain. And actually, we indeed observed the self-association of SR62 tandem valence protein, tandem kinase valence protein, and the kinase 1 domain. They self-associate, but not the kinase 2 domain. So it means that the SR62 tandem kinase protein self-associate while the kinase 1 domain. And this is consistent with this structure prediction, which means the structure predicted is very accurate. And if we put effectors into the prediction, we find that the effectors also bind to the kinase 1 domain, mainly while the beta hairpin area. And this is also a overlapping binding site with the kinase 2 domain. Based on the structures, we did a lot of structure-guided mutagenesis assay, and the mutations disrupted the self-association and the effector binding. So the experiment data prove that the structures are very good and very accurate. Another structure prediction and validation is the interaction between K2 domain and the K1 domain. You can see that they have a very big interface for interaction. So what we did is pick up the residues in the interface on K1 and K2 and make mutations to see if the mutations will disrupt their association and function. First is kinase 1 interface mutations. We introduced several groups for multi-mutations on kinase 1 interface, and all of them showed more or less disrupted the interaction between K1 and K2, these two domains. For example, these two groups of multi-mutations, they totally abolished the interaction between K1 and K2. As you know that the interaction between K1 and K2 is used to inhibit K2's function. So that's why when you infiltrate SR62 tandem kinase protein, the Folens protein with the NLR, it cannot activate the NLR because the K2 is inhibited. But the tandem kinase protein with the kinase 1 interface mutations, they can activate the NLR and cause cell death, which means they become autoactive because they cannot fully interact with K2 and make K2 become escaped, so it can activate the NLR. And then there is the K2 interface mutations. We made 4 groups of multi-mutations on K2 interface, and all of them showed more or less decreased interaction with K1. And we also tested the autoactivity of the kinase Folens protein with K2 interface mutations. None of them showed autoactivity. This is fine. But when we tested the recognition with AVR SR62 effectors, we found that well, the SR62 tandem kinase wild-type protein can recognize the effectors and cause cell death with an NLR. This multi-mutation highlighted in yellow can also recognize the effectors and activate the NLR. This is fine. But these 3 multi-mutations in red, they cannot activate the NLR. And this is because from this co-IP, we can see that these 3 red multi-mutations, they cannot interact with the NLR anymore. And this yellow mutation, which is still functional, still interact with the NLR. So this means these 3 red mutations, they can affect the interaction between K1 and K2, and can also affect the interaction with the NLR. Again, overlapping binding sites. So based on previous results, you can see that the predict structures is pretty accurate. We did a lot of structure-guided mutagencies, and the results are all consistent with the structures. And so take all the results together. A summary for our story is that SR62 tandem kinase protein, if there is no effector, it stays as an inhibited state as a dimer by self-association, and the K2 domain is inhibited by K1 by interaction. And when the AVR62 effector comes, it will bind to the kinase 1 domain by competing for an overlapping binding area with the K2, so it will push K2 away. So the released K2 will expose its NLR interaction interface, so that it can recruit the NLR, interact with it, and activate it, and then activate immune response. So, very short sentence for the SRC2 tandem kinase and NLR pair is recognition and activation is by competition for overlapping binding sites. Thank you, and many thanks to my past and disease group in CSIRO, and special thanks to my supervisor Peter Dodds and our collaborator Brandon Wolf team and the Stephen team from COST. And happy to answer questions. Thank you.
Right, great talk. So we have a question in the chat from Jean. Can the interaction and reduced interactions be recapitulated with purified components?
Sorry, I'm not very sure about what this question means. The reduced interaction between K1 and K2 be resolved with what does the purified component mean?
Like recombinant protein, like can you see it with just the individual proteins, or this was from expressed in the plant, these interactions? Or was
Yeah.
in vitro, I'm saying?
Our experiment is performed by agar infiltration in plants. I don't know if using other
Why
method
am I
can.
asking this question? Because I wonder if there's other things in the plant that are affecting the interactions or interference of interactions, and I wonder if the binding can be demonstrated with purified components, or their interrupted binding could be, then you would be fully explanatory by just those interactions.
I didn't do this experiment, but we have other method and get apart from the plant to hybrid assay, we also perform the co-IP and see observe the similar result. And so I think to purify the components can.
Okay, I'm sorry, we are running out of time, so we should move on to the next speaker.
Thank you.
Back to you, Ashley.
Thank you. All right, so our last speaker of the day is Tianrun Lee, who's a graduate student at the University of California, Davis.
Hi, everybody. My name is Tianrun Lee. You can also call me Jerry. And it's my great honor to have this opportunity to share my story on engineering expanded flagellum perception in plant FLS2 receptors. So just a little bit more intro about myself. I'm a 5.6-year PhD student in Dr. Guido Coker's lab from Department of Plant Pathology at UC Davis. And my primary focus are on specificity and utility of the cell surface pattern recognition receptors, and a little bit on the vector-borne disease biology. All right, so as I mentioned, my research focus on primarily on the cell membrane, where it's the home for those pattern recognition receptors, or PRs. These PRs can recognize conserved microbial features, or called MAPs, from bacteria, fungi, or insects, and mount the immune responses, including the production of reactor altered species, phosphorylation of MAP kinase cascades, and the induction of defense gene expression. And these molecular hallmarks can be tested to determine whether a plant can recognize a certain MAP carrying the receptor. All right, one of the most characterized MAM is called FLAG22 or FLG22. It's a 22 amino acid immunogenic epitope derived from the bacterial flagellum protein. It will need to be digested and released by a host subthalase in the apoplast to be then perceived by a leucine-rich repeat receptor called FLS2. But however, not all bacteria have exactly the same sequence of FLAG22 epitopes. So if you take a look at this phylogeny that's built using the flagellum protein sequence from plant-associated alpha, beta, and gamma proteobacteria, and outside of Pseudomonas aeruginosa species, where is the FLAG22 original described in, you have found that other plant pathogens like Bacterium, Ralstonia, and Xanthomonas, they have a very diverse flagellum protein sequences. And in early literatures, we know that the functional study on FLS2 in Arabidopsis, tomato, or grapevine, they have found a common things is that all those FLS2 homologs can only recognize FLAG22 from Pseudomonas aeruginosa or the other ones that were similar sequence. So they cannot recognize any FLAG22 variants from beta proteobacteria or alpha proteobacteria. That's we call them like canonical perception profile. However, in recent years, there are a few examples FLS2 homologs that can not only recognize Pseudomonas aeruginosa FLAG22 variants, they can also recognize variants from beta and alpha proteobacteria, which including important plant pathogens like Ralstonia or Agrobacterium. So here are the three examples. So we have T. jasminoides or star jasmine from Gentianialis order. We have Vicalis vitis riparia or riverbank grapevine from Vitalis order. Or we have Quercus variabilis or QV from the Ficalis order. And you will see that all those FLS species that host FLS homologs, they are from very distinct and diverse plant orders. And the common thing about them is they can all recognize Agrobacterium FLAG22. So with that information, we basically want to explore two main questions today. Is that what's the recognition profile for those FLS2 variants? Do they just recognize Pseudomonas and Agrobacterium flat22, or is it actually more than that? And second is, can we use that information and to engineer this expanded flat22 perception into other FLS2 via a rational residue swaps? So to answer the first question, of course, we want to have to test the recognition profile with a panel of flat22 variants. So we select 10 different flat22 variants to represent members of the important plant pathogens from alpha, beta, and gamma proteobacteria. And then we use that to challenge the plant, Nicotiana Epithemiana FLS2 mutant plants that are transiently expressing the FLS2 homologs. And then with this combination, or using common immunity assays, including ROS and kinase, to verify whether a receptor can recognize a certain flat22 variants. And here are the lists of the flat22 variants that we tested. Of course, we still have Pseudomonas aeruginosa as the control, and we have Agrobacterium and Rhizobium rhizogenus from alpha proteobacteria. We have Rostonia 2 variants from beta proteobacteria. And we have Dickia, Winia, and other members of Pseudomonas xanthomonas from gamma proteobacteria. If you take a look at this alignment, you'll realize that on the C-terminus, there are a lot of polymorphism C-terminus, and which can be related with their diverse immune perception profile by the FLS2. So here are the results, right? So first, let's take a look at the two very important model plants, Arabidopsis and Nicotiana Epithemiana. And you'll realize that both of them can only recognize Pseudomonas aeruginosa flat22 variants, which is the reason why you're seeing these two bubbles. And of course, the size of bubble also represents the magnitude of the Ross response, which I will show diverse later. While here, I'm showing another species, Solanum persicum of tomato. It can not only recognize Pseudomonas aeruginosa flat22, but it can also recognize Dickia and Winia flat22 variants, but of course with a weaker magnitude. And then we have three examples of FLS2 variants that I just mentioned a while ago. You'll see that they not only can recognize Pseudomonas and Agrobacterium flat22 that they reported, they actually have a much more expanded and diverse recognition profile. One thing worth noting is that TJ and QV FLS2, these two, they can recognize important plant pathogen from Rostonia. So across all those FLS2 homologs, we'll start to wonder what makes them different. Why there are some that can only recognize one of the flat22 in our panel, but some can recognize 6 of them. So what made those FLS2 different? Wondering maybe they're in different colors, or they are so different that some FLS2 start to look like a snake. So to answer this question, we can use generally is an easier way to model those 26 different FLS2 homologs with 2 to 97 different flat22 variants from previous literature backed with real-world perception data. And you will see that no matter how diverse the FLS2 is, they all always interact with the flat22 in a similar manner, which is also clear from the topical view that it's the inner concave surface of the FLS2 that's always interacting with the flat22. So here is our theory. So we know that even though FLS2 with expanded perception and FLS2 with canonical perception have grossly very different amino acid sequence, but it's the LR concave surface region that's regulating the recognition of flat22 and confer differences. So if we want to engineer the perception from FLS2 like QV FLS2, FLS2 XL, we can easily focus on the inner concave surface of the LRs, and that should provide us with the engineered perception if we rationally select the residues to swap. All right, so before we actually do the engineering, there's still one thing missing from the equation. So we know that QV FLS2 is a very powerful FLS2 homolog. It can recognize a lot of flat22, including Estonia, but we still need something that's a backbone, FLS2 that do not perceive those variants to start with. So that's the reason why we searched other close related species, and we found another species called Vagus crenata. So this FC-F+2 shares 87% amino acid identity with QV-F+2, and more importantly, it cannot perceive anything, any flat-22 variants that QV-F+2 can perceive, even if we challenge them with 4 micromolar concentration of the peptides, which make FC-F+2 is a perfect template to work with. So now the first thing we want to do is to make a high-quality structural model of QV-F+2, the co-receptor and B33A, and the Rostonia flat-22 variants, which will be the kind of the target for the engineering approach. And with this model, you can we can select residues by distance and to find the residues in the binding pockets. So that will be the residue of interest. Apart from the structural analysis, we also survey the F+2 sequence in the entire Flagellus order, which including QV and FC F+2. And we want to know the conservation pattern of the entire F+2. So here we use a method called repeat conservation mapping, which will show you across 28 different LLRs of the F+2 and focus on the concave surface region. You will see that there are some region like LLR 10 to 20, you will observe a region with a much darker blue color, indicating they have more polymorphisms and more diversity. We hypothesize that this region of diversity may contribute to the difference of the recognition profile between QV-F+2 and CF+2. And with these two strategies combined, we'll be able to find the residues that we're interested in, and we'll selectively alter them if the alteration will also change the amino acid saturation property, which should be biochemically alter the binding affinity with the flat-22 variants. So here are the results. Like I mentioned, QV-F+2 is very capable of perceiving those flat-22 variants, while FC-F+2 just only perceiving the Pseudomonas aeruginosa flat-22. And for our first synthetic variants, we designed a proof of concept, meaning that we alter the concave surface residues from LR 1 to 28, so all the entire LR domain. Then you will see that this synthetic variant can almost have the same recognition capacity to recognize Rostonia 1, Rostonia 2, and DKF flat-22. And then the next question we want to know is whether the co-receptor is important in this context. So then in the second synthetic variant, we eliminated all the residue change in the binding region that only bind with the co-receptor, that indicating by the LLR 18 to 20. Right, so you'll see from the second synthetic variant, we have suffered a severe reduction of recognition capacity against Rostonia and DKF. So that's kind of not looking good. So that's why for the third synthetic variant, we want to really focus on the diverse region that we identify from the regional conservation mapping. That's from LR 10 to 20, which will generally cover flat-22 C-terminus binding region and some part of the co-receptor binding region. And you will see here from the third variant, we only change 13 amino acids, and that gives us almost the same recognition capacity against Rostonia flat-22. But of course, in a trade-off, we also lose a little bit of recognition capacity with other flat-22 variants. So if we really want to visualize those 13 amino acids on the structure, you'll see that all of them are located in the concave surface of the F+2 LLRs. Specifically, most of them are interacting either with the flat-22 C-terminus or with the co-receptor. And with that, that's to summarize what we did for this project is now we know that the natural F+2 homologs are exhibiting expanded and distinct flat-22 recognition profile. And then now we know that it's possible to engineer expanded flat-22 perception in F+2 homologs via rational residue swaps. And with that, I'd like to acknowledge everybody in the Gita Coco Lab, especially my mentor, and the people that have been involved with this project, especially undergraduate students, and our external collaborator, Daniel, from the Berkeley National Lab, also Andrew Brent from University of Wisconsin, and our funding agency, And with that, I'd like to take any questions if you like. Thank you.
Great talk. Thank you. Anyone has any question? I don't see any yet in the chat. Maybe give like 10 seconds if anyone has any question. If not, we move to the next talk.
I can add. Jerry, nice talk. The decision to use the beach receptor as the template rather than something like you have enough homologs to reconstruct the ancestral sequence. Can you just talk through that decision and how you went about thinking about that?
Oh, right. So, so at that time, our purpose is to find a backbone F+2 that's not perceiving any polymorphic flat-22 variants. So, basically, the rationale at that time is to stay a little bit far away from the Quercus. So all the Quercus species have very, very similar F+2 sequences. So to jump a little bit outside of that genus and find one in the neighboring genus that, but basically we're trying for luck. So and we try to we synthesize the Ficus crenata F+2, and we got lucky. It was indeed not perceiving any polymorphic flat-22 variants. But I think I agree that with all the information we have on hand, their perception profile, it's possible to reconstruct the ancestral sequence and see how that perception profile coming from. So I think it's the interesting aspect.
Does anyone else have any question? Yeah, Adam, please.
Yeah, really nice talk, Chenren. So I'm trying to remember the literature on whether flag 22 peptides get post-translationally modified. So forgive me if I'm mixing up two things, but if that is the case for some pathogens, what are the prospects for sort of structure-guided engineering of FLS2 to recognize such modified peptides? If this is not relevant, just correct me right away. But
yeah, I think
Rainer Vanderhoorn's work, but
yeah, I think some peptides like L15 they need post-translation modification, or other peptides like ROX-X, things like that. But in the case of flat-22, it's generally not the most important thing for perception and the release. So yeah, that's but I think if the PTM is important in this case, I think now with AlphaFold3 you can model peptides with PTMs, and I think that can generally take that into consideration. That's something I can think of.
Right, thanks. I see a comment from Jean. Thank you, Jean.
Yeah, I just remember there were like a series of papers about flagellin modification affecting its function, but I don't know if they specifically said anything about flag 22, but you probably should go look at those papers.
Yeah, Jean, you're right. So they can be post-translation modified, like sugars could be added and then it could no longer be perceived. So potentially there might be some avenues there to maybe re-engineer perception. But I don't think modification has, aside from just cleaving, right, from the intact FLC protein, is important for like normal perception, but maybe, recognition could be resurrected by thinking about, how the plant could modify or the pathogen could modify these components.
Yeah, I think whether the sugars affect release of epitopes could also be possible, but yeah.
All right, if you have any more question, write in the chat box and probably Jerry will answer it. So let's move to the next speaker. Ashley?
Then
Yeah,
the next
so that
speaker
was
is
actually our
me.
last speaker of the day. Yeah, the next speaker is me. There we go.
Right. So Once again, everybody, thank you so much for your participation. We are going to move into our breakout rooms. So as a reminder, these are the rooms, these are the topics of the rooms. Momentarily, they should open up and you will be invited to choose a topic. If at some point you want to switch a room, you can simply click on the breakout room button and you can choose a new room. There will be a member of the IS-MPMI board who's facilitating a discussion in every room. So if you have any questions or if you would like me to put you in a specific room, just hang back. I will be hanging out in this main room the whole time. So if there's any issues, come find me. And everyone
Oh,
should
could
be.
I just make one comment before everyone leaves? If you have any feedback for us about this session, please send it to us. We want to know any way we could modify this format or improve in the future.